Hey everyone, welcome back. So today we're going to talk about enzymes. So for our do now, we asked three questions. Are viruses living? Explain using evidence. Describe the function of the smoothie R, roughy R, and the Golgi. And also what affects populations. We did this to start the class to kind of review some things that we have covered the last 6 weeks because those are very important to kind of keep going back through those. So 3 questions that we're going to talk about today are: what are parts of chemical reactions, how can energy changes be related to chemical reactions, and what is the importance of enzymes in living organisms? That we should be able to answer by the end of the day and no later by the end of the week as we keep practicing what enzymes are. So as we go through and you're laying down at night, you think your body's at rest. However, it's not. You're still digesting food that you ate that day. Your elbow's healing if you scraped it. Muscles and bones are growing and developing. And all these things that happen inside your body are the result of chemical reactions that we're going to talk about today. So kind of get a little intro prior to us talking about enzymes and how they play a role in these chemical reactions. We have to talk about what are chemical reactions and what takes place in chemical reactions. So all living organisms require energy. Energy is the ability to do work, right? And so energy can occur in multiple forms, whether it be electrical, chemical, mechanical, or thermal. And energy can be converted from one form into another. Now, one of the biggest things is talking about thermodynamics. And that energy has to be present in order to be used, okay? And so as we go through, whatever energy is used, we had to have energy in some form or fashion in something else in order for that to occur or to be an equal amount. And so as we go through this might kind of help us go through this topic. So as you guys see this picture before, you guys have looked at where, there's ice, it's melting. That is a physical change. That's not what we're talking about. We're talking about not water staying water. We're talking about water turning into something else. As chemically and making an adjustment to make a different product. And so chemical reactions break or create bonds between atoms, ions, molecules to create new chemical combinations. Now energy is either absorbed or released when bonds are broken and new ones are formed. And so the great way that I remember it is when we add a bond, we're storing energy. When we break a bond, we're releasing energy. And that will sound counterproductive in a couple of slides. However, if we go back to whatever energy we start with is whatever energy we end with, it'll kind of make some sense. So chemical reactions are written as this. We have reactants that make products. So reactants, there's a chemical change to the reactions or the reactants. In this reaction, we follow the arrow and what's left is our products. And some chemical reactions are reversible, meaning that a reaction can proceed in both directions, which means I would see an arrow going this way to the product and an arrow going to the reactants, and in that case it will determine on which way we're going, either from left to right or right to left will determine which one are the reactants and which one are the products. So chemical reactions change substances into different ones by breaking and forming chemical bonds. Reactants are the thing that's changed. Products are what's made within the chemical reaction. And each element within the reaction must be present on each side of the reaction. So we're going to look here. And so let's look at both sides of the equation first. So we have 1 carbon here. We have 1 carbon here. We have 2 oxygen here. And 1 oxygen here, that makes it 3 total. We have 3 oxygen here. We have 2 hydrogen here. We have 2 hydrogen here. So we have to have the same on both product and reactants in order for this to work. Now, bond energy is the amount of energy that breaks a bond. So energy is added to break bonds and energy is released when bonds form, which sounds opposite of what I told you. I told you that when we add a bond, we're storing energy. When I told you we're breaking a bond, we're breaking energy. But on here it says energy is added to break bonds and energy is released when bonds form. Now, going back to thermodynamics, energy has to be from the start to the end. So if I'm adding a bond, or forming a bond to store energy, okay, or to keep energy in, I've got to release energy for the energy level to be the same from start to finish. So this chemical compound, let's say CO2, if I have a carbon, let me draw here real quick, or if I have a carbon molecule plus, or this carbon dioxide molecule plus this water molecule, we're going to create a bond between those two to make H2CO3. Okay? The amount of energy that is on the left side of this equation is going to have to be the same as what's on the right side of the equation. So, if I start out with this on the left side, and I form this bond, okay? But when I add bonds, remember, I'm storing energy, right? In order for me to store energy here, I've got to release energy on the other side for it to be equal, okay? So, activation energy. Activation energy is the amount of energy that needs to be absorbed to start a chemical reaction. So we're going to look at a graph here, and these are my reactants. In order for this chemical reaction to occur, I have to have a certain amount of activation energy. So looking to the right of this picture, I've got to get this ball over the top of the hill in order for it to occur, or to get rolled down the other side. The same thing, this is an all or none. If I do not have enough energy in this chemical reaction, it will not take place. I have to have a certain amount of activation energy, or enough energy to be put into this reaction in order for it to occur in any form. So there are two types of reactions. We have endothermic and exothermic reactions. So the first one is exothermic, and these reactions release more energy than they absorb. The reactants have a higher bond energy than products, and the excess energy is released by the reaction in the form of heat. So, I have my reactants. I have the total energy added, or my activation energy. Okay? I've released more energy than I put in, so I've got to displace energy from somewhere else. In this case, what's left is heat. Okay? That is exothermic reaction. So whatever energy I put in, I've got to have the same out. In this case, in exothermic, I put out too much from what I added, so therefore I've got to release it in some form or fashion known as heat. Now, endothermic. Endothermic reactions absorb more energy than they release. The reactants have a lower bond energy than the products, and the energy must be absorbed by the reaction to make up the difference. So, here are my reactants. Here are my products. I added this much energy. I only released this much. So my products have less energy or bond energy than my reactants. And so in order to make them equal or go through equilibrium, because we have to be the same, we have to reabsorb this energy in order for it to be equal. So now, many chemical reactions help transfer energy in living things, and this transfer involves electrons. And so we have two words. We have oxidation and we have reduction. And so oxidation is when I lose one or more electrons, becoming more positive, because electrons are negative. If I lose a negative charge, I create myself to be more positive. And reduction is when I gain one or more electrons to becoming more negative. So if I'm a positive number, I gain or if I'm a positive 2, I gain 2 electrons, or negative 2. I'm now going to be neutral, which means I became more negative. I went further away from the positivity. And so these don't happen just by themselves. They happen always together. So this is what we call a redox reaction, or an oxidation-reduction reaction. And this occurs when one compound loses electrons and one compound gains electrons. So if you see here to the left, my oxidation is on top, my reduction is on the bottom. I have an oxidizing agent here on the bottom left on the B, and a reducing agent on the top left, because the reducing has 2 electrons. The oxidizing has an even charge. They take the 2 electrons from A, give it to B, which means it's reduced, and then since A gave away its electrons, it's now oxidized, known as a redox reaction. Now, let's get to the good stuff. So catalysts are substances that speed up chemical reactions, and they're going to do this by decreasing the activation energy and increasing reaction rate. So looking at this graph, this big arrow here is activation energy of an uncatalyzed reaction, while the small arrow to the dotted line is activation energy of a catalyzed reaction. So because of this catalyst, I've lowered the amount of energy required because I have help, and because I've lowered the energy, I've also increased the reaction rate. Because now I've created progress a little bit earlier, almost to my end result. And it doesn't have to go as far with my reactants of energy input. So enzymes are catalysts, and they are needed for almost every process, and they are usually made up of proteins. And so enzymes have these awesome things. So if you look to the right here, we have enzyme. Okay, this is sucrase, right? And remember that enzymes end in ASE. We have my active site where these two places are that something will fit. This is my substrate, my sucrose here. My sucrose is going to bind to my enzyme at the active site called the enzyme substrate complex. And in this case, I'm adding water to this molecule to give some electrons, and it's converted into products known as glucose and fructose. So this is my reactant, this is my product, and I added water as part of my chemical reaction for bonds. Now, enzymes are named for their substrates. We have lipase, it speeds up the breakdown of lipose. Amylase speeds up the breakdown of amylose. And so one of the things that also we got to talk about is one that you guys know very well, lactose. Okay, lactose intolerance is a real thing, and it's due to the enzyme lactase. And so as you see this enzyme to the left, how it's folded because it's a protein. Because remember, protein has a certain structure which creates a certain function. It's folded in a certain way to be able to respond to what it needs to. And so lactose intolerance is a variety of symptoms, because some people get really, really sick. Some people could just have like a little upset tummy for a couple of minutes. And so this is due to either how many enzymes or if the enzyme is functioning properly of lactase. Now, you can even go to the store and buy like lactase pills at over the counter. And that will help that if you have some issues with like cheese, that it may help your digestive system, and you usually take it after you eat where the cheese is already in your stomach. You take the pill, and it allows you to start breaking down the lactose. So, enzymes catalyze only specific reactions based on their conformation. Because it's a protein, because it has a certain structure, because it has a certain structure, it's going to have a certain function. In this case, these substrate molecules have to be certain ones. And the molecules in which the enzymes work upon are known as enzymatic substrates. And then these substrates, or what we call reactants, bind to the active site of the enzyme, as you see here to the picture to the right. So now we have the lock and key model, and this helps illustrate how enzymes function. Substrates are bonded together, the bonds of the substrates are weakened to form a product. Now this is very, very specific, because just like if you take the key to your house and try to open up another house, the key will not work because it does not fit the lock. Same thing with enzymes, is if the substrate does not fit the active site of the enzyme, it will not work. And then our last slide. So snake venom has enzymes that break down membranes of the person's red blood cells. So in this case, as you see, the snake bite is right here. This is the reaction to the red blood cells being going through apoptosis or cell death to these enzymes. And so as this goes, you're going to see these enzymes work into the bloodstream, move up the body, so they do the function that they're made to do. And so with antivenom, it's going to block the active site of that enzyme for it to not function. Oh, sorry, this is our last slide. So the last slide is the shape of the enzyme can be altered by certain changes, whether it be ion composition, temperature, or pH. So high body temperatures can cause death because at these temperatures, proteins undergo denature or a change in their 3D dimensional shape. So to the left, we have an active functional protein, and to the right is where we have environmental changes like high temperature, where it begins to unfold. And so this is very, very important because, like for instance, if you have a fever and you take fever-reducing medicine and you still have a high fever for a very long time, you usually go to the hospital to try to, and they will give you IV and things like that to kind of cool you off. And this is due to your brain is made of mostly proteins, and because of a high temperature, you could actually denature some of those proteins in your brain. And so it is very, very important that, with those fevers that we take those seriously, especially that 104, 105 degree fevers. Even those 103 for a long period of time can be some issue. And because it denatures, it will not be able to function and therefore loses its function, which means you lost some proteins that do certain things. Okay, guys. Hopefully that helps you, and I will see you next time. And if you have any questions, please let me know.